We're going to go ahead and get into completing membranes. So we started membranes on Wednesday, and we really just looked at a review. And this was kind of a segue into chapter 4, but you'll find cell structure in chapter 2, I believe. So between chapter 2 and 3, we're really just a review of cell membranes in preparation for getting into chapter 4, okay? And so last time we drew out the cell membrane and we talked about some of the important properties of the cell membrane, that fluid mosaic model, specifically the importance of the phospholipids. The phospholipids creating that bilayer that helps with the selective permeability of that membrane, okay? And so as we get into chapter 4 here, we're really going to be speaking about how transport across that membrane occurs. Now, our membranes are important for allowing transport, so not only movement of material in, but movement of material out, right? Important things need to come in, other important things need to go out. And so it's going to be allowing or facilitating that exchange between the intracellular fluid compartment and the extracellular fluid compartment. So what's happening inside our cells and what's happening sort of everywhere else. It's also selective. So there are some things that can go through and some things that cannot. And this is where we get the separate environment, if you will, in the ECF and ICF. This is why we have more sodium in the ECF, less sodium in the ICF. This is why we have different proportion of different ions in different relationships because of the selective permeability of the membrane. In other words, the intracellular fluid is a different environment. There are different ions, different proportion of molecules than the extracellular fluid compartment because of that selective permeability, okay? Now, the importance of this exchange is sort of twofold here. Oxygen and nutrients need to come in, but then waste products, byproducts, metabolites, CO2 and other metabolites need to be removed from the cell as well. So this is where we get a little bit more into the details of the selective permeability. So this is just the idea that some things can go through that membrane, other things cannot. And this is based on different properties, which we'll speak about, but mainly the non-polar molecules can go through that lipid bilayer. Does anyone remember why or what the relationship is between the lipid bilayer and non-polar molecules or hydrophobic molecules or lipophilic molecules? So we kind of talked about this before, but we spoke about that internal layer, that internal space rather, where we have those non-polar tails of the phospholipids. And so only items or molecules that can exist in that space can go through the membrane, all right? Really important. So things like oxygen, carbon dioxide, fatty acids, some steroids can go through the membrane. And so this is where we get that selective permeability, the amphipathic nature of the phospholipids, only allowing some things through and not allowing other things through. Things like ions, things like polar molecules, large molecules, any type of charged particle normally cannot go through the membrane. They're going to need some type of help, such as a transporter or a channel. And so examples of these things are glucose. Glucose needs a transporter, which we'll speak about coming up. Proteins need some type of transporter or channel. Ions, such as sodium, potassium, calcium, they also need some type of channel, which we usually call ion channels. So really important to distinguish between what can just go right through that membrane via diffusion, or kind of just glide through that non-polar center of the phospholipid bilayer versus other things because of size, because of charge, because of polarity, need some assistance in order to get through the membrane. Okay. So let's go on here to objective 2. This explains the role of the chemical and electrical and electrochemical, really the combination of chemical and electrical, driving force in passive transport, and then how this facilitates substances moving through the membrane. And what we're really doing here is distinguishing between passive and active transport, okay? So let's start by doing just that. So differentiating what is passive transport from active transport. So passive transport is spontaneous movement. It's downhill movement. It's natural, meaning it's going to happen. This is what spontaneous means, without any energy input. And so it's actually going to liberate energy in order to move something passively, okay? And so this example shown here is showing the ball, the orange ball at the top of the hill. In order for this ball to move down the hill, that is a passive mechanism. That is a spontaneous mechanism. That can happen without energy, any energy input. Alternatively, active transport is not spontaneous. It is an uphill movement. It's sort of against the grain. It's going to need energy. So again, think about the balls here. Think about the green ball trying to make its way up to the top of the hill. We're going to need to input some energy to get that ball to the top of this hill. Whereas for passive transport, the orange ball moving to the bottom of this curve is actually going to liberate some energy. It's going to give some energy. It's going to build some momentum as that ball moves passively or spontaneously down the slope here. Okay, so this is the general description or general difference between active and passive transport. Passive transport does not require energy, rather it gives energy and it is spontaneous. Active transport requires energy input, is against the grain, uphill movement that is, again, requiring energy to happen. Okay, so let's begin here differentiating between our two types of forces. So I kind of want to spend a minute here talking about what a driving force is. When we kind of jump into this, we kind of want to explain what a driving force is. A driving force is a force that wants to move a particle, okay? Now we have different driving forces acting across our membranes. In other words, we have forces that are trying to move particles either in or out of our cell across that membrane, okay? Now there are two main types of driving forces that are acting on our cell membranes. There's a chemical driving force and then there's an electrical driving force. And so just kind of giving you a broad overview of what we're going to speak about going forward, we're going to talk about the chemical force separately. We're going to talk about the electrical force separately, and then we're going to sort of combine those two ideas and speak about the electrochemical force. And so the electrochemical force is the combination of these two separate forces that are both acting on our cell membranes, okay? So whereas we're going to sort of split them apart and speak about them individually for clarity, we want to think that they're both equally acting on our cell membranes. And so when we put them together later on, that'll make a little bit more sense in terms of that combined electrochemical force. So again, passive movement is movement down the force. Active movement is movement against this force. So when we're speaking about these chemical and electrical forces, we can have passive or active movement in either of these forces. Okay, so but depending on the spontaneity of that movement, whether it's downhill, whether it's releasing energy, or whether it's uphill and requiring energy, will determine whether it is passive or active for both of these forces. So let's start off with the chemical driving force. Let's describe it here a little bit. The chemical driving force is based upon the concentration gradient, right? The difference between how much particles or solutes are dissolved in one area, let's say inside of the cell, versus how much solutes or particles are dissolved in another concentration outside of the cell. And so we have different concentration of different particles. Let's take sodium, for example. Okay, sodium is at a higher concentration in the extracellular fluid compartment outside the cell and a lower concentration in the intracellular fluid compartment inside the cell. And so that is a chemical force. That's a force that wants to move sodium from high concentration to low concentration, or push sodium across out the membrane into the cell in order to equalize those concentrations, okay? So it's a force that moves particles through the membrane from high concentration to low concentration. And that's the chemical driving force, okay? So let's illustrate that here. Let's illustrate that here on this slide. So we're looking at the chemical driving force, and just to describe or define it here first, this is the movement of molecules or particles down their concentration gradient and so essentially this force wants to move these particles from high to low concentration. Okay. All right, so let's look at a practical example. We have a cell here. Okay, we have two different solute concentrations. Let's start with solute A. So solute A is at a higher concentration on the outside of the cell. So we're going to put more of this solute A outside the cell here. And then at a lower concentration inside the cell. Okay, so solute A, higher concentration outside the cell, lower concentration inside our cell. Let's look at another solute, solute B. Solute B is at a lower concentration outside the cell. So a few particles dissolved out here, and then at a higher concentration inside our cell. So many more of these particles dissolved here. Okay. And so when we think about the chemical driving force in this context, we want to think about what direction of movement each of these solutes would have. And so if we think about solute A, what direction will solute A want to move in? Given that the membrane is permeable, so let's understand first that the membrane is permeable to both our solutes, what direction is solute A going to want to move?
Into the cell?
Into the cell, perfect. So we can draw, our arrow here, showing that directional movement. This is the force, this is the chemical driving force of solute A trying to move it into the cell. Okay. Let's think about solute B. What direction would solute B want to move in? Again, thinking about our membrane being permeable to solute B.
Out of the cell?
Out of the cell, perfect, thank you. So again, moving from high concentration where it is inside the cell to lower concentration where it is outside the cell. This is the movement of solute B. So this is what the chemical driving force is. It's a force that wants to move particles to be at equilibrium. It wants to equalize the concentration on either end or the solute dissolved in the concentration on either end. And so it's going to want to move the particles in order to attain that equilibrium. And that's the force that we're speaking about, that wants to move particles through our cell membrane in order to get them at equilibrium. Okay.